Half a day, everyone. Today we are going to look at advanced photosynthesis. So this is for, I made this video more towards my IB biology higher level students, and that's the level we're going to look at concepts for. First thing we'll look at here is the basic chemical formula for the photosynthetic reaction. We begin with 6 molecules of water, H2O, and 6 molecules of carbon dioxide, CO2. In the presence of light energy, this will become 1 molecule of glucose, C6H12O6, and 6 molecules of oxygen, or O2. In reality, photosynthesis is split up into many different chemical reactions. So really, I just want to stress that this is the overall formula that is happening for photosynthesis, but each of its parts is divided up into many different subsections. Photosynthesis can be divided into 2 major steps: the light-dependent reaction and the light-independent reaction, which is also referred to as the Calvin cycle. Now, in the light-dependent reaction, as you can imply from the title, it requires sunlight or some type of light energy in order for this process to occur. In the light-independent reaction, this process can occur in the presence of sunlight, or it may happen during the nighttime hours when the sun is not out, but it's independent of light being present. It also can be referred to as the Calvin cycle after Melvin Calvin, who was a scientist that was played a key role in discovering the Calvin cycle, those steps of that reaction. The first step of the process of photosynthesis, the light-dependent reaction, occurs in eukaryotic cells within the organelle we call the chloroplast. We're going to identify a few structures of the chloroplast that we will talk about as we discuss the light-dependent reaction. Within the chloroplast, there are lots of different components, as we can see. First, we will note that the chloroplast has a double membrane consisting of an inner membrane and an outer membrane. It's also noted that it contains 70 S ribosomes, so it can perform its own enzyme formation, and it contains starch, which are molecules that are produced at the end of photosynthesis and are therefore sometimes stored within the stroma of the chloroplast. So the chloroplast basic parts, it's got its double outer membrane. Within that double membrane is a liquid that we call stroma, and within that liquid we can find the 70S ribosomes and starch granules. The other key components of the chloroplast are the thylakoid membranes, which are these little round pancake type structures. And if we have a stack of those thylakoid membrane structures together, we refer to that as grana. And within each thylakoid membrane, there is an empty space, a very small empty space, which we refer to as the thylakoid space. Connecting the thylakoid together are structures we refer to as lamella. And that's the basics of the structures of the chloroplast. The first step in the light-dependent reaction is photoactivation. So in photoactivation, we end up with the light coming down and hitting the chlorophyll pigments that are located in photosystem number 2. And photosystem number 2 is going to contain chlorophyll and other accessory pigments. And as the photons from the light come down and hit the photosystem 2, the chlorophyll molecules are going to become excited, and this is going to cause an electron to basically reach a higher energy level, and it's going to be accepted by the primary acceptor, which we call plastiquinone. And this is an electron acceptor, and so the electron will get excited and it will travel through a variety of carrier molecules in the electron transport chain, very similar to how we discussed in cell respiration, with the cell respiration process. So photoactivation. Once the plastiquinone is reduced and the electron enters it, then the chlorophyll becomes a very powerful oxidizing agent. And there's water molecules that are floating around within the inside of the thylakoid space, and those water molecules are going to immediately break up into oxygen and 4 protons and 4 electrons. Those 4 electrons are going to be placed into, and they're going to take up the spot of the ones that were lost in the electron transport chain. The protons are going to be placed within the thylakoid space, and the oxygen gas is going to be released out as a waste product. The electrons that go through the electron carrier, beginning at plastiquinone and then being passed through a variety of electron carriers, as that process is happening, just like in cell respiration, we end up with protons being taken in from the outer stroma of the chloroplast, and they are brought into the thylakoid space. So those protons drawn in from the electron transport chain, along with the protons that are generated from photolysis, are going to make a big proton gradient on the inside of the thylakoid space. Next, we have the process of chemiosmosis, which is very similar again to the cell respiration process. The proton gradient that develops within the thylakoid space will cause a concentration gradient, which will allow then for the protons to move through the ATP synthase and out back into the stroma. And as that proton is brought through the ATP synthase, this is going to have adenine diphosphate plus inorganic phosphate turn into ATP, which we know is our energy molecule, which will be used by the chloroplast in the next cycle. Next, we have an additional photoactivation. This time we are activating photosystem number 1. And so the light energy is going to come down and hit the chlorophyll molecules in photosystem number 1. And that is again going to pass that energy on to chlorophyll molecules in the reaction center, which raises an electron to a higher level. And this process called photoactivation. So as this electron passes along the carriers within photosystem 1, the end will be ferredoxin, a protein that's located outside the thylakoid, and then this will join to a molecule called NADP reductase. And what that is going to do, it is going to form reduced NADP. And this is an electron carrier molecule just like the NAD from cell respiration. And this reduced NADP is going to be utilized in the second part of the photosynthetic pathway. So looking at our products of the light-dependent reaction, we're harnessing the energy and we're taking that energy and we are going to be making ATP and reduced NADP. These molecules are formed and are within the stroma of the chloroplast. And there they are going to go on to fuel the next part of photosynthesis, the Calvin cycle, or we also call it the light-independent reaction. The light-independent reaction takes place within the stroma of the chloroplasts in eukaryotic cells, and it consists of three main types of reactions. The first set of reactions is referred to as carbon fixation. The second set is referred to as reduction, and then the third we refer to as regeneration. And we are going to go through each of those steps now. Looking at the first step, carbon fixation, within the stroma of the chloroplast, there is lots of enzyme which we call ribisco. The full name is ribulose bisphosphate carboxylase, but we just refer to it as ribisco. And this is arguably one of the most important enzymes, as well as one of the most as far as numbers goes, one of the most that has the most in the world of this particular enzyme because of its importance within the carbon fixation step of photosynthesis. So what we have is we have a molecule, a 5-carbon molecule called ribulose bisphosphate, which we abbreviate as RUBP, and that's what it's usually referred to as. So this molecule, RUBP, is also located within the stroma. So the CO2, which the plant gets from the atmosphere, comes together with the RUBP, which is a 5-carbon sugar, in the presence of ribisco, and ribisco will catalyze the reaction of joining these two molecules together. As soon as that happens, it becomes a 6-carbon molecule, which immediately will become 2 molecules of glycerate 3-phosphate. So it's actually going to end up being a 3-carbon molecule, which we call glycerate 3-phosphate. And that is the carbon fixation step of photosynthesis. The next part of the Calvin cycle, or the light-independent reaction, is the reduction phase. So in this phase, we take the glycerate 3-phosphate molecules, which, remember, there were 2 of them generated in the carbon fixation state. And those glycerate 3-phosphate molecules will utilize the ATP that was synthesized in the light-dependent reaction and the reduced NADPH, which was also synthesized in the light-dependent reaction, and it will convert the glycerate 3-phosphate into what we call triose phosphate. So for every 2 glycerate 3-phosphates that happen, each of these will be doubled, and we will end up with 2 triose phosphates at the end of this reduction phase. In the third step of the Calvin cycle, or the light-independent reaction, we have our triose phosphate that was formed at the end of the reduction phase, and that triose phosphate has 2 different possible roles. One of those roles is some of the triose phosphate is going to be utilized to make our glucose molecules, our end product, which will actually probably end up becoming a long chain, a polymer, which we refer to as starch. Now, some of those other triose phosphates are going to again utilize more ATP that was generated in the light-dependent reaction, and we are going to form more RUBP, which will then fuel our next set of carbon fixations, and will keep this cycle going in a cyclic pattern. So to briefly summarize the Calvin cycle, we have RUBP and carbon dioxide, which are coming together in the presence of the enzyme ribisco, and they are going to generate glycerate 3-phosphate. That glycerate 3-phosphate, in the presence of ATP and reduced NADP that was formed in the light-dependent reaction, are going to reduce the glycerate 3-phosphate and form triose phosphate. Once that triose phosphate is formed, Some of that triose phosphate will be regenerated with the help of ATP to become more RUBP so that this cycle can continue to go round and round. Some of this triose phosphate will be made into other carbohydrate molecules, and we usually refer to it as glucose, but that isn't always the product. That triose phosphate may be made into starches or other types of carbohydrates as needed by the plant. And basically, for every one molecule of glucose that's formed, we need 6 cycles of the Calvin cycle for that one glucose, because every time, one time it's going through, it's adding in one carbon molecule is being fixed. So if our glucose contains 6 carbons, that means we need 6 cycles in order for the glucose to be formed. To summarize the chloroplast again, we have the structure, this organelle, which is located in photosynthetic organisms, primarily plants, some bacteria. And the chloroplast contains a chloroplast envelope, which consists of an outer membrane and an inner membrane. It has a dual membrane. That dual membrane helps to keep the required enzymes and products necessary for photosynthesis in a contained environment. Within the chloroplast, we have thylakoid membranes, which are very important because they contain the pigment chlorophyll, which is a key component in photosynthesis, in the absorption of the photons from the sun. And those thylakoid membranes, inside they have a space, and that's important for the proton gradient to happen for the ATP synthase and ATP production. Stacks of thylakoid membranes are called granum. So this would be a granum or grana in plural. Those are going to be filled throughout the chloroplast in improving the amount of surface area for these chemical reactions to take place, which, as we know, surface area is very key for our biological reactions. Within the stroma of the chloroplast contain a variety of enzymes that are necessary for the Calvin cycle. It also contains 70S ribosomes, which are important for making the necessary proteins, the enzymes that are necessary for these chemical processes to occur. And there is some naked DNA scattered throughout the stroma, which contains the codes necessary for those enzymes and proteins. Usually we'll also see a starch molecule within the chloroplast, and that is from the product of the Calvin cycle, where it will store the excess carbohydrates as they are formed as a product of photosynthesis. And on occasions we'll see some lipid droplets that are there. And that pretty much summarizes our chloroplast. To briefly summarize photosynthesis, it consists of 2 major parts: the light-dependent reaction. The light-dependent reaction takes the photons from sun or other types of light energy, and it's going to harvest it with the help of electrons, electron carriers, electron transport chain, photosystems 2 and photosystems 1, and proton gradient chemiosmosis, and it is going to make ATP and reduced NADP, and these are going to be used later on for the second part of the photosynthetic pathway. Now, in the second part, we call this the light-independent reaction because it can happen at any time of day, and light can be present or not present. It doesn't really matter. In the light-independent reaction, we are going to use carbon dioxide. We're going to fix it with RUBP, and through a series of reductions, we are going to end up with triose phosphate, and that triose phosphate is going to eventually lead to our carbohydrates, which will fuel the cell respiration process, which is necessary for ATP production. The process of photosynthesis also does occur in prokaryotic cells. We primarily just looked at the eukaryotic cell method. Now, with prokaryotes, they obviously do not have the chloroplast, but they are still able to photosynthesize, and their chlorophyll is located within the cell membrane, and they can still carry out these chemical reactions and still produce energy. As well as carbohydrate molecules. It's a little bit less efficient of a process because everything isn't contained in one area, but it does the job that's necessary for the single-cell prokaryotic organisms to survive. So that's pretty much a summary of photosynthesis, and have a great day.